Hello, everybody, and welcome to this last seminar of 2021. My name is Patrice Brassard, and I'm a professor at Université Laval, and I'm co-organizing this seminar series with my colleague, Dr. Kathleen Miller from University of Texas. Maybe just we'll revise one last time the rule of engagement, following a little bit of information about the CARNET, which has been established in 2010, and this network is a multidisciplinary community of researchers working on various aspects of cerebral blood flow regulation. And if you would like to join our network, just email your CV to Dr. Records, and you have the email address here. In terms of a rule of engagement, you should know this by now, but again, just a quick review. So please keep your microphone on mute and video on throughout the session. There will be time for questions at the end of the presentations, and usually people have written their question in the chat box. So and we will read each question to the presenter and the session chair will then invite you to ask your question if you raise your hand. And finally, the session will be recorded and posted on the CARNET website and YouTube within the next week. So we are really happy to have Dr. Margie Davenport as the keynote speaker today. She is an associate professor at the Faculty of Kinesiology, Sport and Recreation, as well as the director of the Program for Pregnancy and Postpartum Health at the University of Alberta, Canada. And her talk will be about cerebral blood flow and pregnancy.
All right, so thank you so much for the opportunity to be able to speak in this seminar series. I'm especially pleased to be able to showcase the work of four really outstanding scientists, early career scientists. And over the next 30 minutes, I'll be just going over an overview of the impact of pregnancy on the brain to really be able to set you up for their work. So I'm currently located at the University of Alberta up in Edmonton. It's a beautiful campus. If you ever have the opportunity to visit, I do encourage you to do so. In the winter, there's great skiing. In the summer, lots of trails to go and visit. But probably one of the best parts of the University of Alberta, at least in my very biased opinion, is our research center here. So I'm an exercise physiologist. I investigate the impact of physical activity on the lifelong health of pregnant and postpartum women and their children. This is our research center, the Program for Pregnancy and Postpartum Health. We have a fabulous research-only gym, but we also have a cardiovascular suite where we can do assessments of the brain, the heart, and as well as peripheral blood vessels. So to give you a brief outline, I'm going to go over some of the physiological adaptations to pregnancy. If you're not familiar, there are a lot of them over the course of pregnancy. Briefly touch on structural changes to the brain, then talk about cerebral blood flow, but I'll try not to skip any of the subsequent presentations. Talk a little bit about complications during pregnancy, the potential impact of physical activity, and then we get to the really good stuff, the introductions and our trainee presentations. So if you aren't already aware, pregnancy is associated with some really rapid and profound physiological adaptations that are really designed to support the needs of the growing fetus. So pregnancy is about 40 weeks from the last menstrual period right up until birth. Term is considered to be equal to or greater than 37 weeks gestation. And as pregnancy progresses, estrogen, progesterone, and the placental hormones are really going to rise within the maternal circulation and affect virtually every system of the woman's body. The 40 weeks of pregnancy, you're going to hear us talking about different trimesters as we go along. If you're not familiar, the first trimester is from conception to 13 weeks. The second trimester is 14 to 27 weeks, and then the third trimester is 28 to 40 weeks. And so pregnancy is, again, a really unique time in a woman's life. There's lots and lots of adaptations that occur. They occur in virtually every system within the body. Some of the ones that I'm most interested in are looking at metabolism. So there's going to be vast changes in metabolism, including about a 20% increase in resting VO2. The respiratory system has some of the greatest adaptations and most noticeable adaptations. You're going to see about a 30 to 50% increase in resting ventilation. Arterial CO2 is going to drop. Bicarbonate is also going to drop. Chemoreflex sensitivity, all really relevant to adaptations to the brain. A lot of my work is looking at the cardiovascular adaptations to pregnancy. And again, there are some really profound changes. Keep in mind, this all happens over a 9-month period. So you're getting about a 20% increase in heart rate at rest. Blood volume, believe it or not, goes up by about 50%. So as a result, you're going to have approximately 30% increase in cardiac output. But vascular resistance over the course of pregnancy actually drops so that you end up having a relatively maintained blood pressure, although some studies show that there is a decrease in the second trimester. But interestingly, the pregnant brain is actually often forgotten. So we know a lot less about the impact of pregnancy on the brain. We know that early in pregnancy, estradiol and progesterone are really going to begin to rise quickly and by term reach levels that are not seen at any other point outside of pregnancy. And in fact, a pregnant individual is going to produce more estrogen during one pregnancy than throughout the rest of her lifetime when she's not pregnant. And so this is really important because these sex hormones, estradiol, progesterone, have been shown to regulate neuroplasticity, neuroinflammation, and behavior as well. So I want to begin just by briefly touching on some of the changes in cerebral structures. So for a long time, whether we were talking about the cardiovascular adaptations or the brain adaptations, we just assumed that pregnancy was a fairly benign period in a woman's life. For a long time, we assumed that pregnancy had little to no impact on the brain. But there have been a number of studies that have been coming out over the last couple of years, which have really challenged this idea. So one of the recent ones that came out just in 2016, so we commonly assume that the brain of the mother is basically the same as somebody who is not pregnant or has never been pregnant. However, in 2016, there was an MRI study which followed first-time mothers from pre to post-pregnancy, as well as 2 years follow-up. So we have here on the y-axis, you have change in gray matter volume on the y-axis, and then the time in months across the x-axis. So we have here, before pregnancy, they did MRI to measure gray matter volume. The blue shaded area is going to be pregnancy. And then they also did a measurement at post-pregnancy, so in the postpartum period, as well as a 2-year follow-up. And what they found is that from sorry, my mouse isn't quite working, from pre to post-pregnancy, that there was a drop in gray matter volume that occurred, and then there was no rebound in the postpartum period. So this reduction actually persisted for 2 years following delivery. There was a really recent study following up an additional 6 years postpartum. And so again, you have gray matter change on the y-axis, and then you have pre-pregnancy, the blue shaded area is pregnancy itself, postpartum, and then on the far right side of the axis is going to be 6 years postpartum. So once again, you can see in pregnancy that there's a drop in gray matter volume. And following that, after postpartum to 6 years postpartum, there's an age-related decline in gray matter volume. The light gray line here, that is actually individuals who have never been pregnant. And you can see that there is a normative age-related decline in gray matter volume that's mirrored here. But the important thing here is that these changes that we're seeing in pregnancy, the reduction in gray matter volume, is actually persisting. So is this an issue? Well, the really easy answer is that we don't actually know. So when we think about gray matter loss, we actually think of it as being quite detrimental, such as what we would see in older age. So as we get older, there's going to be a reduction in brain volume. However, there is actually precedence for seeing gray matter volume in healthy younger individuals. So for example, during adolescence, there's also a surge of hormones where we see a reduction in brain volume. But this is actually believed to occur as a result of widespread pruning of synapses to actually make the brain more efficient. And so the authors are suggesting that perhaps pregnancy might actually represent another time in a woman's life, similar to puberty, where hormonal fluctuations may actually affect the organizational structure of the brain. Now, the other interesting thing about this particular study was that the areas of the brain that shrunk the most were actually involved with social cognition, which is a person's ability to figure out what a person is thinking or feeling. And they also found that the reduction in gray matter actually predicted maternal attachment. So the reduction in blood volume or brain volume that we're seeing with pregnancy may potentially be a positive adaptation to actually help support the mother in taking care of her baby. Regardless, these data and others are actually really starting to demonstrate there's a persisting effect of pregnancy on the brain. But we're really here to talk about cerebral blood flow. Although we're gaining a better and greater appreciation of the acute and persisting structural adaptations in the brain, I would say that less is known about cerebral blood flow during pregnancy, and we certainly have a less of a consistent understanding, as you'll see in my as well as subsequent presentations. So I just want to highlight again the multitude of physiological adaptations that occur during pregnancy, specifically related to the brain. Over the course of pregnancy, we see changes in cardiac output, changes in blood pressure, certainly changes in arterial CO2, as well as blood volume. And all of these we would expect are going to challenge blood flow regulation. However, early work in this area really does present conflicting results. So before 2015, relatively little was known about the normative adaptations of cerebrovascular reactivity and cerebral blood flow during pregnancy. And there really was only one study that looked at ICA, but not specifically MCA or other cerebral vessels, looking at the longitudinal changes in reactivity or blood flow across all of pregnancy. And so we were pretty fortunate early on in the lab to have a very dedicated pregnant woman who came in pre-pregnancy to be assessed in her early follicular and luteal phases, as well as weekly throughout pregnancy. Overall, what we found was that the middle cerebral blood flow velocity was fairly stable across pregnancy. So we have these closed bars here, pre-pregnancy, first, second, and third trimester. You can see the p-value was, getting close, although it certainly wasn't significant, but generally it was fairly stable, the resting cerebral velocity across the 3 trimesters of pregnancy. These data are supported by a handful of cross-sectional studies which examine the impact of pregnancy on brain blood flow in the late 2nd and 3rd trimester using MRI. Other studies looking at transcranial Doppler ultrasound, which is what we used, suggested that cerebral blood flow in the middle and posterior cerebral artery is either unchanged or slightly lower in pregnancy compared to non-pregnant women. Now, I do want to highlight, though, that these data are mostly using transcranial Doppler ultrasound. As many of you will be aware, the cerebral vessel's diameter may not be consistent during pregnancy or in response to changes in CO2. But to this point, we don't have any specific information about that. And it's definitely an area that needs to be addressed moving forward. We also looked at cerebrovascular reactivity to CO2 hyperoxic rebreathe. And our case study, so we have here, the cerebrovascular reactivity to CO2 at 5 weeks gestation, so very early pregnancy. She was just barely pregnant, about 3 weeks pregnant, and then late pregnancy, so just before term here as well, looking at cerebral blood flow velocity or cerebral velocity and end tidal CO2. You can see that weeks 5 compared to weeks 36, that the reactivity was actually increase substantially by the end of pregnancy. And also, if you take a look at panel C, again, we have the cerebral reactivity to CO2 going from the follicular and luteal non-pregnant phase right up to term. There is an increase in the cerebrovascular reactivity that increases over time. And so we suggested that based on this literature that the progressive augmentation in cerebral reactivity to CO2 across gestation suggests that there might be an improved cerebral reserve, which is inversely related to stroke risk. Now, I just want to touch on one thing. So one of the issues that we run into with pregnancy is that many of our studies are quite non-invasive and we don't often have blood samples or other measures that go along with it. So for those of you who are considering doing work in pregnancy in the future, I really advocate to try and look at this so that we can better understand some of the mechanistic changes. The complexities of the physiology of pregnant women are such that I think it's really necessary to gain a strong understanding. So in this particular case, again, we know that pregnancy is associated with very well-characterized increases in ventilation and associated chronic respiratory alkalosis. This is primarily thought to be a result of increased progesterone. Now, in non-pregnant populations, this chronic respiratory alkalosis, such as what you would see at altitude, is associated with an increased or augmented cerebral reactivity. Under these conditions, altered acid-base regulation may actually influence cerebral regulation. And so what we're seeing is that as we went across pregnancy, so we have the follicular and luteal phases here, and moving across gestation, coming down here, so this was at 38 weeks gestation, that we actually saw that there are reductions in bicarbonate because of the metabolic compensation during the chronic respiratory alkalosis, which can limit the buffering capacity during subsequent acute increases in carbon dioxide. And that this could potentially augment reactivity. So for a variety of other reasons, the increased cerebrovascular reactivity during pregnancy might actually be due to changes in the stimulus that are mediated by altered acid-base regulation as opposed to vascular adaptation per se. But certainly we don't know enough about this at this point in time. And I also want to highlight that the findings of our case study are actually quite in contrast to other studies which compare CO2 reactivity between pregnant and non-pregnant individuals, where they found no difference between groups. Again, many of these were not longitudinal, and certainly much more work needs to be done. And some of this newer work is actually going to be presented in our early career researcher presentations that are upcoming. So I'm quite excited for you to see a bit more on that. So when we talk about autoregulation in pregnancy, with the exception of some upcoming presentations, information on autoregulation during pregnancy are extremely scarce. There's only a couple of studies, but based on animal studies primarily conducted out of Marilyn Skow's group, suggests that cerebral autoregulation may actually be improved. But again, this is in rodent pregnancies. There are 2 available human studies prior to those that are being presented subsequently, which show that either autoregulation is either unchanged or it's improved slightly in human pregnancy. It's important to note that the studies use very different methods to assess autoregulation. They use different gestational ages. They didn't account for potentially confounding variables such as parity or lifestyle. So certainly, again, if you are considering doing work in pregnancy in the brain, I would strongly encourage you to do so. It's a wide open field and lots needs to be done for sure. And neurovascular coupling. Again, there's extremely limited information available at this point in time. The one study, with the exception of those that are upcoming, only one study found impaired neurovascular coupling in women with gestational diabetes compared to normal glycemic pregnancy. However, with the exception of those that are upcoming, there are no comparisons with non-pregnant individuals. And so just as a quick overview, certainly the presentations that are coming later this morning are going to provide more information about our understanding of cerebrovascular regulation in healthy pregnancy. But our current understanding or idea based on mostly Doppler ultrasound work is that middle and posterior cerebral artery blood velocity decreases during pregnancy, and CO2 reactivity and autoregulation are either maintained or potentially slightly increased with pregnancy. But again, I would like to emphasize that more detailed investigations, including measurement of blood vessel caliber, blood and expired gas analysis, certainly sex hormones, as well as looking at investigations across gestation, are really badly needed. So why does this all matter? Well, there are a variety of different reasons. First off, the physiological changes of pregnancy have been suggested to actually alter cognition. And so I won't talk about it very well at all during this presentation, but there's also emerging work which is suggesting that the pregnancy-related changes in the brain may also influence the risk of depression and anxiety as well. And so that's a really emerging area of research. But one of the reasons that I started working in the area of pregnancy in the brain was based on my own experiences of extreme forgetfulness during pregnancy. When I went into the literature, there was actually relatively little information. Most suggested that it was more lack of sleep or distraction than anything else. But if you talk to pregnant women, this pregnancy-induced brain fog or baby brain really refers to this subjective decline in cognition that's reported by up to 81% of individuals who have been pregnant. Forgetfulness certainly is most commonly reported. Lots of people will experience forgetting appointments, whether they turned off the stove, which happened to me many times, where they left their car, which still happens to me. And most of these symptoms are really quite mild, for sure. Likely only obvious to those who are close to them, likely are not going to impact with work and daily living, unless of course you leave your stove or forget where your car is altogether. And from personal experience, it really is more of a nuisance than anything else. But studies evaluating the objective measures of cognition have illustrated some decline, while others have suggested that cognition is not actually altered. And so these people have long suggested that these feelings are really more related to a lack of sleep, general fatigue, or as I mentioned, I was told a lot by my physician that it was just plain old distraction, which may have entirely been the case. But regardless, our understanding of the pregnant brain is surprisingly limited. However, there was a recent meta-analysis that was published back in 2018, which looked at the impact of pregnancy on cognitive impairment using objective measures. And they found that pregnant individuals had a small but significant decline in cognition. There were only at this point in time 20 studies reporting on 709 pregnant and 521 non-pregnant participants across a variety of domains. In this particular case, you can see that there was actually only six studies reporting. So this is again a very highly understudied area. But they illustrated a small effect size showing a decline in general cognitive function across pregnancy. They also they looked at a variety again of different areas of cognition, but they also found a moderate effect size showing a reduction in memory function during pregnancy that can actually appear as early as the first trimester, believe it or not. So again, the effects are generally quite mild. Awareness and potentially intervention in more severe cases to help maintain cognition and memory during pregnancy might be helpful. And certainly based on some of my postdoctoral fellowship work with Dr. Marc Poulain, looking at the effects of exercise on cognition, I will be advocating that perhaps exercise might be quite beneficial. And so, so just to give it a summary, so this was published by Elisa Gallia's group a couple of years ago. I added just a couple of small additions to it. But in general, what we see in late pregnancy, certainly very high circulating estradiol and progesterone, some reduction in verbal memory and other measures of cognition, reduction in overall brain volume. We're seeing in general a reduction in middle cerebral artery velocity and PCA velocity as well. There should be an increase in autoregulation, not a decrease, please ignore that, and likely no change in reactivity. Certainly less is known about the postpartum impact. We expect that there's some maintenance in cognition. Some are actually suggesting there might be even improvements in cognition, but we do know that gray matter appears to be reduced over the longer term. And going into middle age, well, we don't know yet. Now, all of the data I've presented has been really in uncomplicated pregnancy, but we do want to know about how the brain is impacted by complications that arise during pregnancy as well. So not all pregnancies are going to progress smoothly. We know that 85% of women will become pregnant in their lifetime, and 20% of these women will develop complications during pregnancy that impact their current and future cardiovascular and cerebrovascular health. So the one that I'm going to focus on right now is hypertensive disorders of pregnancy, because of the potentially detrimental impact of these disorders on maternal brain. So these disorders include preeclampsia, gestational hypertension, and chronic hypertension. Although each of them are very distinct in how they're diagnosed and characterized, they are all characterized by high blood pressure during pregnancy. And they occur in about 7 to 10% of pregnancies in Canada and the US, and are a leading cause of maternal fetal morbidity and mortality. So certainly cerebrovascular complications such as stroke are extremely rare during pregnancy. These are very young, healthy women. However, the incident rates are 3 times higher in pregnancy than in non-pregnant women. And we've been seeing an increase in the risk of stroke of about 50 to 80% over the past 20 years. 18% of strokes in young women occur during pregnancy, at delivery, and then actually many more occur over in the early postpartum period, and are very much related to the development of preeclampsia. When we talk about preeclampsia in the brain, just as it's relevant to a future presentation this morning. So preeclampsia is new onset hypertension after 20 weeks gestation, with proteinuria or other signs of end organ damage. It's established that preeclampsia has some peripheral vascular dysfunction. And when we talk specifically about the brain, there's increased cerebral blood flow, reduced autoregulation, and cerebrovascular reactivity to CO2. Now, why do we care about pregnancy and pregnancy complications? Well, the vast majority of women will go through pregnancy and they can adapt to the changes of pregnancy quite easily. However, there is a subset of women who have underlying risk factors for cardiovascular disease. In these small subset of women, they are going to present with either gestational hypertension or preeclampsia during pregnancy. However, following delivery, this condition disappears. They might be at increased risk later on in life. But these women are at actually much increased risk of developing cardiovascular and cerebrovascular disease earlier in life than we would expect otherwise. And so this has been a really active area of investigation, looking at the longer term effects of pregnancy complications. This meta-analysis was published just a couple of years ago. And what they found was that hypertensive disorders of pregnancy increase the risk of cerebrovascular morbidity by nearly 300%. These are otherwise young, healthy women that are having a very elevated risk of developing cardiovascular and cerebrovascular disease. Now, we don't know a lot yet about the lifelong impact of preeclampsia on the brain. Dr. Jill Barnes and Dr. Kathleen Miller have really been leading the way on this. So I'm very excited that Dr. Miller is going to be presenting. She's the last presenter actually this morning. However, We do know, and we now recognize that pregnancy unmasks future chronic disease risk, and that the evidence is so strong that the American Heart Association listed pregnancy complications, including gestational diabetes and preeclampsia, as risk factors as strong as smoking for the future development of chronic diseases, including stroke. However, the question is, can we prevent these diseases from developing? And if we can we actually reduce their long-term impact in terms of risk for cerebrovascular and cardiovascular disease? So these are conditions. Preeclampsia is a condition that affects about 10,000 women in Canada. 18,000 pregnant women are affected with gestational hypertension, and 21,000 are affected with gestational diabetes or diagnosed with gestational diabetes in Canada. And so again, we've been looking at potential preventative measures over the last number of years. As you will see in a subsequent presentation, we believe that there's a key role of physical activity in preventing these pregnancy complications. So several guidelines now around the world, including the American Heart Association, Hypertension Canada, the UK Chief Medical Officer's Physical Activity Guidelines, the American College of Obstetricians and Gynecologists, all encourage engagement in physical activity to reduce the risk of developing hypertensive complications during pregnancy. Now, this is important and relevant for those individuals who develop pregnancy complications. If we're able to prevent the development of preeclampsia during pregnancy, perhaps we might actually be able to reduce or delay the development of these cardiovascular and cerebrovascular complications later in pregnancy. So here in Canada, we published the 2019 Canadian Guideline for Physical Activity throughout pregnancy. We encourage 150 minutes of moderate intensity physical activity spread over 3 or more days of the week to derive clinically meaningful benefits. And this was based on a series of systematic reviews that we did that Dr. Skow was involved with, where we found that prenatal exercise did actually reduce the odds of developing gestational diabetes, gestational hypertension, and preeclampsia by about 40%. So the impact of physical activity during pregnancy on the risk of these complications are quite significant. However, we might understand the impact of exercise on the development of these chronic conditions, but we actually know very little about the impact of exercise on the brain. And so back in 2016, Dr. Broussard's group put out a call to action, basically, urging the world, the research world, to start to address and understand the acute and chronic impacts of prenatal exercise. So on the left-hand side of this diagram, we have the normal pregnancy adaptations that we would expect to see in pregnancy. At that point in time, there was no information about the impact of exercise on the brain. And so they derived this information from non-pregnant populations. But we do now have a couple of studies. 3 will be talking in subsequent presentations to be the first 3 studies to actually start to address this. So we're quite excited for sure. Again, in terms of the field specifically, there are major gaps that are remaining in this particular field. I like to sum it up by, if you think of a gap, that's probably one. We're currently addressing the impact of high intensity exercise on the brain. It's really important to start looking at things like resistance training and sedentary behavior, as well as sleep in the brain during pregnancy. Are we able to actually prevent and adapt, prevent cerebrovascular complications in those individuals who are at high risk for developing preeclampsia? Will we actually alter their brain function potentially? There have been some suggestion in non-pregnant populations that perhaps exercise can increase brain volume. Whether this can also happen or attenuate the pregnancy-related decline in brain volume that we're seeing, who knows? And many, many different complicated populations, twin pregnancies and type 1 diabetes. Again, you name it, needs to be done. So I would strongly encourage each of you, if you're considering doing work in pregnancy in the brain, that your help is very much needed. And so I'll just wrap it up. I'm going a bit over time, apologize. I'll wrap it up by really thanking my trainees. They are the ones that do all of the work. You're going to see some of them present their work over the next little bit. They're the rising stars, they're the bright people. I need coffee to stay awake most times and think clearly. And so they certainly make things work around the lab. If there are any questions, I'd be happy to take them before we get into the really good stuff with our early career presentations.
Thank you very much, Margie, for a fascinating story. So do not hesitate to ask, include your questions in the chat. We have already 2. So Dr. Panray, are the reductions in gray matter volume caused by hypocapnia or changes in intracranial pressure? Recent work has shown that brain matter can be reduced by increases in ICP.
I think, again, the easy answer is we don't know. My understanding, reading their work, that they don't know yet. Those were the first 2 studies. They were actually looking at the same cohort of individuals. And so at this point in time, we don't know. Just the fact that they're even showing it is really quite fascinating.
Question from Melissa Wright. Is baby brain also reported in women who don't keep the baby after pregnancy, such as like surrogate?
Yeah, so they were demonstrating that baby brain can actually occur as early as the first trimester. And so, yeah, we would expect, even if there is fetal demise, that there would potentially be some effects of pregnancy brain, but certainly we would also be somewhat dependent on how long the women did carry the pregnancy.
Any other questions? I may have 1 or 2. Do we have data about, because you discussed a little bit about the autoregulation, neurovascular coupling. We know that cardiac output may have direct and indirect influence on cerebral blood flow changes. Do we have data about like whether cardiac output is as important as like non-pregnant individuals for cerebral blood flow? So
studies have definitely included measures of cardiac output, but I don't think that I have read anything, and certainly our work has not related cardiac output to changes in the brain. It'd be easy enough to look at though.
Mm-hmm. Okay. Thanks. A question from Austin Davis. What are the long-term consequences of reduced gray matter into the postpartum period, and could this be related to postpartum depression?
Great question. Again, this is such a new area. That second paper actually just only came out earlier this year. I don't think we understand the long-term consequences at all. So certainly when we see things like there's a reduction in gray matter, that we immediately think that is quite detrimental. But the authors are actually suggesting, because of the high surges in sex hormones, again, we see surges in sex hormones in adolescence, and this is actually more of a reorganization of the brain synapses to make it more efficient. Possibly that's the same thing happening, or it's also possible that it's actually detrimental, and that greater reductions in brain volume might be linked to depression. But from what I've seen, there isn't any data on that yet, but I could be wrong.
Thank you very much. Any other questions? If not, I will let you present the next speaker. Thank you again.
Okay, perfect. So I am really, really pleased to be able to introduce 4 exceptional scientists that have been really advancing our understanding of the impact of pregnancy on the brain. I'm especially pleased and relieved that all 4 of them agreed to have the work showcased in this seminar series. And so the first one that I'll be introducing is Brittany Matenchuk. So Brittany is a PhD student at the Program for Pregnancy and Postpartum Health. She's currently doing her PhD. She's not looking at the brain, but she is looking at pregnancy. So the work she's presenting on today is work that she did as a research assistant with myself and Dr. Craig Steinbach a couple of years ago. And her presentation's entitled A Longitudinal Study of Cerebral Blood Flow Regulation During Pregnancy. So Brittany, the floor is yours.
Thank you so much, Margie. Yeah, so as Margie said, I'm going to be talking about cerebral blood flow regulation during exercise in pregnancy. And I'm a new PhD student with the Program for Pregnancy and Postpartum Health. So we know that there's an increased cardiovascular load and circulating factors that rise over the course of pregnancy, and they pose a unique challenge to cerebral blood flow regulation, as Margie discussed. But the longitudinal changes in cerebral function in pregnancy have not been described. So namely, blood volume increases by 50% during pregnancy, and this leads to significant increases in cardiac output and heart rate. Additionally, progesterone causes a higher sensitivity to carbon dioxide and a decreased threshold of the central ventilatory chemoreflex response to carbon dioxide. This results in lower maternal arterial CO2, which facilitates the placental CO2 transfer. During this study, the objective was to examine the longitudinal changes in neurovascular coupling, cerebrovascular reactivity to carbon dioxide, and exercise across the non-pregnant and pregnant state. And we hypothesized that resting cerebral blood flow would be similar or slightly decreased over the 3 trimesters of pregnancy compared to non-pregnant women. And we further hypothesized that the reactivity of the middle and posterior cerebral arteries, MCA and PCA, to visual stimulation, CO2 rebreathe, and exercise would be increased from the 1st to the 3rd trimester compared to non-pregnant controls. For this study, we collected 47 total participants who were over 18, non-smokers, and were having a singleton pregnancy and were not pregnant over the last 6 months for the non-pregnant women, and free from known neurological, respiratory, or cardiovascular diseases. And they completed testing at 1 or more time points. So collecting data from individuals across preconception through pregnancy can be methodologically tricky. So as a result, we collected data from 2 or more time points on 22 of the individuals in our 47. And for 6 of those, we had measures in both the non-pregnant and at least 1 time point in pregnancy. All participants provided both verbal and written informed consent prior to participation in this study, and gestational age was calculated from the first day of the last menstrual period and confirmed by ultrasound. None of the participants reported a history of gestational diabetes, gestational hypertension, or preeclampsia because we know that those could be related to cerebrovascular function. Non-pregnant women were tested during the early follicular phase of the menstrual cycle, with the exception of those who were taking hormonal contraceptives. As you can see here, we instrumented participants with a transcranial Doppler ultrasound to collect blood flow velocity of the middle and posterior cerebral arteries, so this helmet device here. And we used a pneumotachometer, spirometer amplifier, and gas analyzer to collect inspiratory and expiratory gases throughout testing. So with this mouthpiece device. And we also collected continuous ECG and blood pressure during testing. For the visual stimulation test, following a 2-minute eyes closed baseline in a dark room, the participants would open their eyes for 5 trials of 30 seconds of flashing light at 0.1 seconds of light dark cycles, separated by 4 periods of 30 seconds with eyes closed in a dark room. For the modified Duffin hyperoxic rebreathe test, following 5 minutes of baseline, participants voluntarily hyperventilated until their end tidal CO2 was reduced to about 25 torr for 1 minute. After a full inspiration and expiration, participants were then switched using a 3-way valve to a bag containing 95% oxygen and 5% carbon dioxide, and instructed to inspire 1 deep breath. Participants then resumed natural breathing until their end tidal CO2 reached 55 torr, or the bag deflated, or they used previously agreed upon hand signals to indicate that they wish to terminate the test. For the incremental exercise to volitional fatigue, which is just when the participant was maximally fatigued, we had participants sit for a 5-minute inactive baseline, and then they cycled for 5 minutes at 25 watts, maintaining a pedal rate of 50 RPM as a warm-up. Following this warm-up, the pedal rate was maintained at the 50 RPM, but the work rate was increased by 25 watts per minute until the participant reached volitional fatigue. We collected their perceived exertion and recorded every second increment during exercise using a Borg scale. And when they reached volitional fatigue, they then had a 5-minute cool down period. These are the descriptive characteristics of the participants, and due to the unbalanced overlap between participants, we used cluster analysis and multi-level modeling to account for the decreased variability within participants across their own measures. Data are presented here as means plus standard deviations, and from this point forward, I'll use the following terminology for the different groups, where NP represents non-pregnant, TM1 is first trimester of pregnancy, TM2 is second trimester, and TM3 is third trimester. There were 26 individuals in the non-pregnant group, 13 in trimester 1, 21 in trimester 2, and 20 in trimester 3. There were no differences in age, height, or pre-pregnant BMI of the participants. However, the gestational age and weight increased with advancing pregnancy, as we would expect. Also, parity was not different between these groups. And these are their hemodynamic and respiratory characteristics. So as expected, the baseline mean arterial pressure or blood pressure was lower while heart rate was higher across the pregnant groups compared to non-pregnant. End tidal CO2 was lower in TM2 and TM3 compared to non-pregnant, and tidal volume was significantly higher in trimester 2 and trimester 3, while the VE was significantly increased in trimester 3 only. Respiratory rate was not significantly different in pregnancy. At rest, MCA velocity was progressively lower in trimester 2 and trimester 3 compared to non-pregnant, and PCA velocity reached a nadir or decreased in second trimester, but then was similar in trimester 1 and trimester 3 to the non-pregnant values. MCA and PCA resistance and conductance were not different in trimester 1, 2, or 3 compared to non-pregnant at rest. And the MCA and PCA resistances are calculated as blood pressure overflow, while the MCA and PCA conductance is a calculation of flow over blood pressure. So these are taking into account concurrent changes in blood pressure. These are the MCA and PCA responses to the neurovascular coupling or visual stimulation test. So I'm going to go through a few graphs like these. So I'm just going to take a little while to explain what's going on here. So on the x-axis, we can see the duration of the stimulus, and this is the data averaged over the 5 visual stimulation periods. The y-axis in order is our MCA velocity, PCA velocity, MCA conductance or blood flow over pressure, and PCA conductance, which is the same. And the legend for the groups is at the bottom. So blue represents the non-pregnant, red is first trimester, green is second trimester, and yellow is third trimester. The shading represents the 95% confidence interval, and all values were adjusted for the similarity between unbalanced repeated measures within each participant. MCA and PCA mean and conductance were not different between the groups, but there is a notation here because the PCA had a shorter time to peak velocity in the second trimester compared to non-pregnant. So you can see here that the green might have reached this peak value at a sooner time than the blue line here. And moving on to the modified Duffin rebreathe test, there was no difference between groups for the response in MCA or PCA during the test. This is the MCA response to incremental exercise to volitional fatigue. And so on the bottom here, we have the watts, which is the power output during the exercise test. And so 0 starting at that rest period and then increasing to volitional fatigue, which was different for each participant. Not everybody reached this 175 watts. And as we can see, there was a drop in the MCA during progressive exercise in the non-pregnants, which this is what we've observed in other non-pregnant or general population groups. But we didn't observe this drop in the pregnant groups. And we were wondering, what's causing this? So then we looked at the MCA corrected for the end-tidal CO2 during the exercise test and the reactivity to this value from the Duffin hypercapnic rebreathe test. And we found that this explained the difference in the MCA during the exercise test. We then also looked at PCA during the exercise test, and the response in PCA was actually similar between the pregnant and non-pregnant participants. And interestingly, when we corrected for the individual carbon dioxide reactivity from the rebreathe test and the change in end-tidal CO2, this actually didn't explain the PCA response to exercise in the pregnant groups. So in summary, we found that MCA was lower in trimester 2 and trimester 3 at rest compared to non-pregnant. The MCA was lower in non-pregnant, but not pregnancy during peak exercise. And this seemed to be explained by the end-tidal CO2 during the exercise test. And afterwards, we found that there was no difference between values. The PCA was lower in trimester 2 compared to non-pregnant at rest. And overall, there were no differences in the neurovascular coupling test and cerebrovascular reactivity between the pregnant and non-pregnant groups. In conclusion, brain blood flow did not appear to be significantly changed during pregnancy. It simply responded to the changes in blood gases that were occurring. So I would like to thank my lab and the support of Dr. Margie Davenport and Dr. Craig Steinbach and all of my co-authors on the paper that came out of this amazing work and all of our amazing lab members. Thank you so much.
Okay, great job, Brittany. So it looks like there is one question currently from Dr. Broussard, which is a two-parter. So the first one, in regards to resting hemodynamics, what could explain the different behavior of the PCA at trimester 2 versus trimester 3?
I'm not sure if I totally know. I would assume there might be a difference in sample size. Could be explaining that. Yeah, I don't know, Margie, if you have any more insights.
No, I don't think we do at this point in time.
Yeah.
We've struggled with that one for quite a long time. I think this is one that really does need follow up with more mechanistic assessments, whether it's like the arterial blood gases. I also think, and same thing with most Doppler studies, that we also need to start looking a lot more about vessel diameter. I do think that's likely playing a role. We just haven't had the ability, I guess, to do that yet, but we are working on it. The second question from Dr. Broussard is for CO2 reactivity. Did you measure blood pressure during CO2 rebreathing? And if so, any increase in blood pressure during this test?
Ooh. So during it, we did measure. I don't exactly remember, but if I stop share, I can go double check.
Yeah, so if memory serves, there was a small increase in blood pressure during the CO2 reactivity. Dr. Timko has a question for you. He was asking if the relationship between end tidal CO2 and arterial CO2 is similar between pregnant and non-pregnant.
Ooh, I would assume that it is, but I haven't checked. Yeah, I would assume that there is a relationship there. Yeah, I don't know. Dr. Timko, do you know if there's a relationship there?
Yeah. Yeah, we have some of that data. I'm not sure. Nothing's coming to mind where there has been a comparison. I would be surprised if somebody has not looked at it before, but yeah, off the top of my head, Dr. Timko, I don't know. Any other questions? Okay, so Brittany, you are off the hook. Thank you so much. And next up, it is my absolute pleasure to introduce our next speaker, Dr. Áine Brislane. So Dr. Brislane is currently a postdoctoral fellow at the Program for Pregnancy and Postpartum Health, working on a large variety of projects, including the impact of physical activity on health outcomes in twin pregnancy and maternal heart disease. She will be presenting today a very, very small portion of her PhD work. She had a very large project, very exciting project from her PhD work at Liverpool John Moores University under the supervision of Dr. Nicola Hopkins and Dr. Helen Jones. And these data represent some of the very first data reporting on cerebrovascular function in response to exercise training. So Dr. Brislane.
Okay, thanks Margie for the introductions. So yeah, I'm just going to present some of the data that I collected during my PhD. So my talk is going to be on the effect of aerobic exercise during pregnancy on prenatal cerebrovascular function. So a little bit of an overlap, I guess, between some of the data that's been presented this morning. So far, really, as Margie alluded to, the influence of exercise on cerebrovascular function is largely understudied. And this is despite the potential for abnormal or exaggerated responses to exercise to be indicative of cerebrovascular complications in pregnancy. And really worryingly, I guess the only remedy for cerebrovascular complications in pregnancy is premature delivery. And that alone has consequences for both mom and baby. So really, this is like a very important topic and something we should be all concerned with. So what happens to cerebrovascular function during pregnancy? So some longitudinal data shows that MCAV, or middle cerebral artery velocity, decreases longitudinally across pregnancy. We also have some data on the circulation at the posterior cerebral artery. So negligible changes throughout pregnancy. Towards the end, we can actually see an increase, but in general, it's lower than the non-pregnant state. And the posterior circulation is of particular interest during pregnancy because it's subject to cerebral edema in pregnancies that are complicated by hypertensive disorders. Hopefully this graph looks familiar. So I'm just going to comment on cerebrovascular reactivity here. So this is what Margie had shown earlier on. So they were really the first study, which is a case study, to illustrate that cerebrovascular reactivity increases across gestation. And that's illustrated here with the open circles. So overall, pregnancy appears to exert some kind of influence on cerebrovascular parameters. However, our knowledge about whether exercise can augment or exacerbate these responses is yet to be discovered. So the aim of, part of the aim of my PhD was to investigate the effect of a 6-month exercise intervention during pregnancy on prenatal cerebrovascular function. So we recruited healthy pregnant women through social media and clinical contacts at the end of their first trimester. Participants self-selected to be in a control or exercise group, and they came to the laboratory on 3 separate occasions, and we tried to keep these as close to the end of the first, second, and third trimester, and that's abbreviated as T1, T2, and T3. After the first visit, the exercise group took part in a partially supervised exercise intervention, completely revolved around aerobic exercise. And at the time of the study, this was in line with the Royal College of Obstetrics and Gynecology prenatal exercise guidelines. And you can see here in the table what that intervention comprised of in terms of the number of sessions completed, total weeks of training, etc. So the ladies were supervised on 1 session per week and then did the rest remotely, but we would monitor heart rate using polar beat and polar flow. So for the cerebrovascular tests, participants were instrumented with a 3-lead ECG, a phenometer, and a transcranial Doppler ultrasound headband. And we also had a mouthpiece to monitor PET CO2. The middle and posterior cerebral arteries were insunated for the neurovascular coupling test. So this image here is just from a lab chart file. So for the neurovascular coupling test, participants completed a 5-minute sequence of opening and closing, so represented by the O and the C, opening and closing their eyes and looking at a visual stimulus on an iPad. The data was collected with the lab chart, and from the channels you can see what we were quantifying. So CO2, posterior cerebral artery velocity, middle cerebral, and MAP, so mean arterial pressure, and PET CO2. So from the channel you can kind of see with the posterior cerebral artery like a slight fluctuation compared to a pretty stable response at the MCA. For the cerebrovascular reactivity test, we used a hypercapnic protocol, so similar to what Brittany had used. So this comprised of a 1-minute baseline, voluntary hyperventilation until we reduced PET CO2, and then onto a gas mix for 3 minutes. So this was 5% CO2, 21% oxygen, and balanced with nitrogen in this Douglas bag here. And during that test, we also monitored the response at the common carotid artery using ultrasound. Relative MCAV was then plotted against PET CO2, and cerebrovascular reactivity, or abbreviated to CVR, was quantified by linear regression. Some other outcomes that we quantified as well was VO2 peak, and we did that by adopting or implementing an Astrand rhyming protocol. And we also monitored physical activity and sedentary behavior for 7 days after each measurement. Our outcomes of interest then were analysed using a linear mixed model. So for the results, I guess similar to what Brittany had shown, first of all, I guess none of the, or both groups were similar on entry to the study in terms of age, body mass, body fat, VO2 peak, and blood pressure. And the only parameter we saw to change across the pregnancy was body mass and subsequently BMI. So this was represented by a main effect for time, a significant main effect for time. In terms of the activity data, so the sedentary behavior outcome, this was lower for the exercise group. So the exercise group appeared to be spending less time being sedentary. But despite the exercise intervention, the physical activity levels remained similar between the exercise and the control group. In terms of the cerebrovascular outcomes, so as expected and in line with previous research, we observed a reduction in MCAV across the pregnancy, and this remained similar between the exercise and control group, so just a main effect for time. In our study, the PCAV, or the posterior cerebral artery velocity, did not change across the pregnancy with no effect for time. However, there was an intervention effect. And just by looking at the data, it appeared that the exercise group seemed to maintain stable levels of PCAV across the pregnancy, whereas the control group had a slight decrease in their PCAV. In terms of the neurovascular coupling response, we didn't see any time intervention or interaction effect here. And with the CVR test, so the rebreathe, we didn't see any time intervention or interaction effect here. So PET CO2, carotid artery diameter, both absolute and relative CVR were the same, and the R value, or the R squared, with the CVR test. So from our study then, the MCAV reduction is similar to what is observed in other studies. And this may be due to a number of factors. We're still kind of, I guess, on the edge of trying to understand the mechanisms behind these changes. But it may be related to vasodilation downstream from the vessels, from the cerebrovascular vessels, potentially related to respiratory changes during pregnancy, which would impact CO2 levels, and then some speculation that sex hormones might play a role in these changes at the cerebrovascular blood vessels. We observed a potential exercise intervention effect on the PCAV. It's important to note here that the exercise group did actually have a slightly higher, although not significant, starting point with the PCAV, so at the end of trimester 1. Nonetheless, it's still a very interesting finding, especially when we think about the posterior circulation being implemented in cerebral edema and hypertensive disorders. So certainly something that we'll want to explore further. And in relation to the CVR test, so we didn't see any change across pregnancy, which is slightly different to what Steinbeck, King, and Davenport have shown. Perhaps that's related to the sample size. Theirs was a case study. We had a cohort. So this is something that we'll be able to resolve further in future studies. It's also a different response. So in non-pregnant cohorts, research has shown that CVR can be increased with exercise training. So our results are in conflict with that. And it's plausible that we just need a larger sample size for this study. So we had 18 in total, 11 in the control and 7 in the exercise group. And our NVC response, so we didn't see anything in terms of gestational age or exercise training. And this is consistent with non-pregnancy literature. But as I mentioned, it may be a more valuable assessment in pregnancies complicated by hypertensive disorders. So to conclude, MCAV is reduced in pregnancy, while the PCAV may be influenced by exercise training. The NVC response and CVR are unchanged with pregnancy, with or without exercise. And future research may look to confirm these and compare against pregnancies that are experiencing some kind of complication. Just to acknowledge my PhD supervisors, so Dr. Hopkins, Professor Helen Jones, and Dr. David Lowe, and for Sophie for helping in the lab, also to the one-to-one midwives and the Liverpool Women's NHS Trust for helping us recruit participants. And any questions, happy to take them.
Okay, great job, Dr. Brislane. Anybody have any questions for her? Okay, so Dr. Broussard had a question. Did you include cerebral autoregulation data in this study?
Yes. I wasn't allowed to present it.
We're only given 10 minutes for each of these presentations, so.
Yeah, so we did measure cerebral autoregulation. So we found some trends in the data with improving cerebral autoregulation, but I think we needed a larger sample size. So we ran this was a pilot study, and we did run some sample size calculations. So to be able to detect differences, we'd need 33 participants in both groups. So that's something we'll probably be looking to do. And I think Rachel will comment on cerebral autoregulation as well.
Great. Any other questions? So another question from Dr. Broussard. Any idea why MCAV and PCAV behave differently?
I think it comes back to kind of what you said about we don't really know structurally like what's happening, the diameters at those points. Also with the sex hormones, so there are sex steroid hormone receptors on cerebrovascular, the vascular beds. So we don't really know enough about kind of the activation of those, whether the MCAV has got more receptors than around the PCAV, and whether that impacts and increased sex hormones, so estrogen during pregnancy, whether one vascular bed is more responsive to those. That's, I think, a little further down the line in terms of understanding those kind of things. But yeah, I think, yeah, it's definitely an interesting question. So I guess the simple answer is I don't know, but we can speculate maybe what's happening. So maybe the MCAV or the MCA is a larger blood vessel at the cerebrovascular bed. So potentially it's experiencing some kind of dilation or not. So there's tons of like structural things that we really don't know that could be impacting the behavior of both vessels.
Okay, that's great. Thank you very much. So we'll be moving on to our next presenter. So I'm very pleased to introduce Dr. Rachel Skow. Dr. Skow is a postdoctoral fellow at the University of Texas at Arlington, and she's presenting on some newly published results from her PhD work at the Program for Pregnancy and Postpartum Health with myself and Dr. Craig Steinbach. Her presentation is going to cover the impact of acute and chronic exercise on dynamic cerebral autoregulation from her PhD trial. So Dr. Skow.
Okay, so Margie's taking the pleasure of introducing me. I just want to thank you, Margie, as well as Dr. Rickards and Broussard for hosting this. I'm so excited that we're finally talking about pregnancy in the brain. So yeah, I'm going to be sharing some work that I did during my PhD as part of a large randomized controlled trial that I ran in my PhD, actually at the same time as Áine, although I don't think we really knew that we were doing the same thing at the same time at that point. So I'm going to start by thanking Margie and Áine and Brittany for providing a lot of the background and rationale for my study. I will keep my background brief as a result. So throughout my PhD, I was very interested in the role of exercise on cardiovascular health and pregnancy. And specifically, one of the aspects that interested me was how exercise might impact cerebral blood flow regulation in pregnancy. And indeed, Dr. Broussard and colleagues published this lovely review back in 2016, highlighting that the research on pregnancy and brain blood adaptations is an important call for action within our research community. So it's really no surprise to me to see the abundance of new and novel research being presented here today from around the world. And so what I think that is really important as regards to exercise training and cerebral vascular health, and why it's important to consider is because pregnant women are recommended to exercise throughout their pregnancies. And so as everyone has really established so far, that this is the importance, and that there might be evidence for altered cerebral hemodynamics during pregnancy that could be impacted by physical activity. So really relevant to what Dr. Davenport mentioned for my talk is that we know the dynamic cerebral autoregulation, so the regulation of brain blood flow by blood pressure, can be impaired in pregnancies that are affected by high blood pressure. And so given that prenatal exercise can decrease the preeclampsia risk, we postulated that it could affect dynamic cerebral autoregulation as well. And so the aims of my study were to determine whether or not dynamic cerebral autoregulation was affected immediately following acute exercise, and also to determine the effects of aerobic exercise intervention on dynamic cerebral autoregulation. And I hypothesized that acute exercise would not impact dynamic cerebral autoregulation in a healthy normotensive pregnancy, but that an aerobic exercise intervention could improve dynamic cerebral autoregulation. And so in order to test this, I performed a large randomized control trial called the PEACH study. And so the data I'm presenting to you today are just a small part of this. And so here in this trial, I was investigating the role of prenatal exercise intervention on several facets of cardiovascular health that included the cerebral circulation and its regulation. And so participants were enrolled into this trial if they were less than 18 years of age, or sorry, more than 18 years old, less than 20 weeks pregnant, and pregnant with a single baby. All of the women in my study were self-reported inactive at enrollment, and they all had their healthcare providers sign off on the PARMEDx for pregnancy physical activity readiness form. And this indicated that they had none of the absolute contraindications to exercise. None of the participants were taking any medications that could affect any of our outcome measures. Importantly, for this is that no participant in this study had any history of either recent or recurrent syncope. And so in total, over 3.5 years, I randomized 59 pregnant participants into either exercise or control conditions, and 51 completed the study. And so in accordance with the consort guidelines for reporting and randomized control trials, the data I'll be presenting to you today is an analysis of all available data from any outcome from any participant who was randomized. So our protocol involved having pregnant participants come in their second trimester between 16 and 20 weeks for a cerebral blood flow assessment, where we continuously measured heart rate, blood pressure, and middle cerebral artery velocity. First, we performed an assessment of dynamic cerebral autoregulation using a single sit to stand. So this involved having 5 minutes of sitting, followed by 3 minutes of standing. And we evaluated dynamic cerebral autoregulation using standard methods. And this was completed by Laurence Lebrecq in the Brossard lab. And she was blinded to study a group for the analysis. So following that first sit to stand, participants performed a symptom-limited peak exercise test, which consisted of a 5-minute warm-up, followed by incremental exercise, increasing in intensity every 1 minute until volitional fatigue, followed by a 5-minute cool down. So this protocol is identical to the one that Brittany used in her study. And then after the cool down, we performed a second sit to stand trial. So following this initial assessment, participants were randomized into either exercise or control conditions. And those in the exercise group were prescribed aerobic exercise at 50 to 70% of their heart rate reserve for up to 40 minutes per session on 3 to 4 days of the week. And at least one of these sessions was supervised by me. Then all participants returned in the third trimester between 34 and 36 weeks for a follow-up assessment that was identical to the first. So first, I'm going to show you the results from our resting hemodynamics. So we have the control group on the left and the exercise group on the right, with the second trimester and third trimester data representing the pre and post exercise intervention or exercise training. And the data here were analyzed using a 2-factor mixed effects model. And as shown by the mean effective gestational age, we show some of the normative and hopefully expected hemodynamic responses to healthy pregnancy, which include rise in heart rate and blood pressure and a decrease in middle cerebral artery occurring between the second and third trimester. Importantly, this table also shows that the rise in heart rate during gestation was attenuated in our exercise group. And this highlights the efficacy of our exercise intervention. So for the rest of the data from the sit to stance that I'll be showing to you today, it will be presented in a similar format. So I'm just going to take a moment to orient you to the graphs in general. So on our y-axis is our outcome of interest. And on the x-axis, we have our pre-exercise or first sit to stand. And then we have the post-exercise, which is post-acute exercise, or the second sit to stand. The control group is shown by the black circles and the exercise group by the white, and the data is plotted as mean and standard deviation. So here we have the second trimester pre-intervention time point, and then on the right will be the post-intervention or trimester 3 time point. So all this data was, we completed the statistics for these using a 3-factor mixed effects model. And so this allowed us to determine the main effects of gestational age, group, which represents our intervention, and then acute exercise, which is the pre to post exercise, as well as the interaction between the 3. So here what I'm showing is the resting mean arterial pressure during the seated period prior to standing. And so we have a mean effect of acute exercise such that blood pressure is higher in the post-exercise period. But importantly, no participant experienced any post-exercise hypotension or symptoms of presyncope during this resting period following exercise. And then on the right here, we have the resting middle cerebral artery velocity, where we show a mean effect of gestational age. So that expected decrease in middle cerebral artery velocity in the 3rd trimester. And then also that the resting blood velocity was lower in the post-exercise period, possibly due to an increase in ventilation. Here, there were no effects of the exercise intervention on either of these measures. So next, looking at the sit to stand trials. So during the transition from sit to stand, this is the drop in mean arterial blood pressure that's occurring. So here we have the results showing that the drop in blood pressure upon standing was less in the 3rd trimester and was greater in the post-exercise period. But importantly, because blood pressure was higher in the post-exercise period, there's actually no difference in the absolute blood pressure occurring upon standing in either sit to stand. So here we have the decrease in middle cerebral artery velocity. And similar, we have a larger decrease in the post-exercise period. And this is not impacted by gestational age, potentially due to the effects of gestational age on resting middle cerebral artery velocity. Again, for both of these, there was no effect of the intervention on this response. So next, we're showing the time delay for the onset of the regulatory change, which is the time lapse between the beginning of the sit to stand and the increase in cerebral vascular conductance index. So taking into account both blood velocity and blood pressure. And so here there was no effect of gestational age, group, or acute exercise on the onset of the regulatory response in this study. Similarly, there was no effect of age, group, or gestational age group or acute exercise on the rate of regulation, which represents the speed at which this regulatory response is occurring. But here we saw a 3-way interaction. And this is because there was an improvement in rate of regulation in the post-exercise period, post-intervention in the exercise group only. And so this potentially suggests an improvement in dynamic cerebral autoregulation with training. But we interpret this with caution, as it is confounded by the seemingly opposite effects of training observed in the control group at the pre-acute exercise time point. So this might be due to the variability in the measurement in the study, the smaller sample size that we had for some of these measures, but also possibly due to a complicated interaction between acute and chronic exercise that we have yet to determine. So just in conclusion, these data really highlight the adaptability of the cerebral circulation in pregnancy, and that in healthy pregnancy, the brain is able to accommodate the large changes that are simultaneously occurring within the cardiovascular system. We do believe these data show potential to see improvement with exercise training. And so similar to Áine, it might have just been limited by the smaller sample size in this study. I definitely didn't have 30 people in each group. Regardless, though, these data show the safety of performing peak exercise tests in pregnant participants, providing that a warm-up, cool down, and a resting period are completed. And last but not least, in line with everything you've heard today, I think this just emphasizes how much more work we have to do with all areas with respect to blood flow regulation in pregnancy. So I'm sure you all have tons of ideas for studies now. So I'd like to thank you all for being here today. So this work was just recently published in the Journal of Applied Physiology and is a collaborative project with collaboration between the University of Alberta and the University of Laval. and Dr. Broussard's research group. And so if you have any questions, I can take those now, but you can also reach out to me on email or on Twitter.
Okay, great job. Thank you, Rachel. Does anybody have any pressing questions for Rachel? Okay, so Dr. Timko has a question for you. Was the drop in MAP during the DCA test to stand similar between pregnant and non-pregnant and pre to post exercise?
Thanks, Dr. Timko. So I had no non-pregnant people in this study, so I can't comment on your first aspect of your question, unfortunately. The decrease in mean arterial pressure was larger post-exercise. So there was a greater drop in blood pressure after an acute exercise, so our exercise test, but their blood pressure started at a higher point. So they actually didn't drop to a lower blood pressure value in terms of absolute blood pressures.
I have a question for you, Rachel.
Okay.
So the intervention was based on previous guidelines, and as you're well aware, the exercise guidelines for pregnancy have actually recently been updated. Do you think that increasing either the intensity or the duration of the intervention would have altered any of your results?
That's always the question. So yes, I started this project before we put out the new guidelines. So our intervention started like Áine's with 3 sessions at 15 minutes of moderate intensity exercise, plus a warm up and a cool down, so 25 minutes of exercise, and built up over about 8 or 9 weeks to 30 minutes 3 times a week. With the warm up and cool down was about 40 minutes 3 times a week, so 120 minutes a week of moderate intensity exercise, which is pretty close to the guidelines. I think from a clinical perspective, we know that less can still be beneficial. So as little as about 45 minutes of exercise can reduce the risk of gestational hypertension, for example. But also that there's kind of a dose volume relationship. So more can be better. So really just, I guess, a great question for another future grad student to be able to address if they want to.
Okay, great. Thanks very much.
Thank you.
And so finally, we're on to our final speaker for this morning. I'm really excited to introduce Dr. Kathleen Miller. Dr. Miller is a research associate at the University of Wisconsin-Madison in the Barnes Lab. She and Dr. Barnes have really pioneered our understanding of the longer-term impact of hypertensive disorders of pregnancy on the brain. And she'll be presenting on her recent work describing the impact of hypertensive pregnancy history, capacity of response to isometric hand grip exercise. So, Dr. Miller.
All right, well, I want to thank you so much, Dr. Davenport, for that introduction. And also thank you to the organizers. This has really been a pleasure attending these seminars. So today I'm going to be talking about some of the long-term effects of a hypertensive pregnancy, specifically on cerebrovascular function. So as Dr. Davenport introduced, preeclampsia is a hypertensive disorder of pregnancy characterized by de novo hypertension and proteinuria after 20 weeks of gestation, and it occurs in approximately 8% of pregnancies. During a normal pregnancy or a normotensive pregnancy, muscle sympathetic nerve activity goes up 50 to 100% compared with pre-pregnancy levels. However, in a preeclamptic pregnancy, muscle sympathetic nerve activity as well as pressor responses to sympathoexcitatory stimuli are augmented. So preeclampsia has been described as a disorder of sympathetic hyperreactivity. Now, the long-term effects or postpartum effects of preeclampsia are still not entirely clear. History of preeclampsia is associated with developing hypertension during midlife. And based on large epidemiological cohorts, a history of preeclampsia is associated with a higher risk of dementia and cerebrovascular disease. So despite this, the mechanisms by which a history of preeclampsia may elicit these long-term impacts on brain health are unclear, including the long-term impact of preeclampsia on cerebrovascular health. As I mentioned previously, preeclamptic pregnancy is associated with sympathetic hyperreactivity during pregnancy, and it was unclear if this persists long-term. However, in a study of postmenopausal women, so tested 35 years later postpartum, it appeared that sympathetic responsiveness to an isometric handgrip, which is a sympathoexcitatory stimulus, did not differ by pregnancy history. Yet it's possible that there are divergent cerebrovascular responses to the stimulus based on pregnancy history. So the objective of this study was to determine if the cerebral blood velocity response to a sympathoexcitatory stimulus differs by pregnancy history in postmenopausal women. And I wanted to mention that the present study is an ancillary study to a larger project conducted at the Mayo Clinic Specialized Center of Research Excellence on Sex Differences with the PI, Dr. Virginia Miller. And so the objective of the Mayo Clinic score was to determine how hypertensive pregnancy disorders, natural menopause, and menopausal hormone therapy may affect risk for cardiovascular disease and cognitive decline. So these studies included many comprehensive assessments of cardiovascular and brain health. So for the present study, the participants were recruited using the medical record linkage system of the Rochester Epidemiology Project. And these participants identified as women, and they were pregnant between 1976 and 1982. They all gave birth in Olmsted County, Minnesota, USA, with a confirmed history of either preeclampsia or a normotensive pregnancy based on the medical record. Then at the time of the current study, which was again about 35 years post-pregnancy, participants must have also currently been a resident of Olmsted County, Minnesota, or surrounding areas. They were non-smoking, non-obese, not on hormone replacement therapy, and had no underlying disease with the exception of controlled hypertension. So the participants arrived to the laboratory after a 4-hour fast and no exercise, alcohol, or caffeine for the last 24 hours. And they lied supine while they were instrumented with a transcranial Doppler ultrasound to measure velocity through the middle cerebral artery and a phonometer to measure beat by beat blood pressure. And they were also instrumented with a blood pressure cuff on their upper left arm that would be used for the post-exercise ischemia. Baseline measurements were recorded, and then the women were asked to perform an isometric hand grip exercise with their left arm at 30% of their calculated max voluntary contraction until fatigue. At fatigue, the blood pressure cuff on their arm was then inflated to 220 millimeters of mercury, and that remained inflated for 90 seconds, which was the post-exercise ischemia part of the protocol. After the 90 seconds, the blood pressure cuff was deflated, and then the participants rested quietly for 2 minutes for a recovery period. Throughout the entire protocol, middle cerebral artery velocity, mean arterial pressure, and heart rate were recorded continuously. So here are the characteristics of our participants. We had 27 postmenopausal women with a history of a normotensive pregnancy, or NP, and we had 21 women with a history of preeclampsia, or abbreviated as PE. By design, the women were similar in age and the amount of years since pregnancy. There was also no significant difference in blood pressures between the two groups. However, those with a history of preeclampsia did have a higher incidence of taking antihypertensive medications. Those with a history of preeclampsia also had a lower middle cerebral artery velocity at rest compared with those who had a history of a normotensive pregnancy. So here are our blood pressure and cerebral blood velocity results. So the x-axis shows the protocol baseline, And then the first third of the hand grip exercise, the second third of the hand grip exercise, and the final third. And this was all relative to each participant's time to fatigue. The PEI portion is the post-exercise ischemia, and that's when we have the cuff on, occluding the arm. And then the final portion is the REC, which is the recovery period, and that was the 2 minutes after the cuff was deflated. So women with a history of a normotensive pregnancy are shown in blue, and those with a history of preeclampsia are shown in red. On the faint y-axis, you can see the blood pressure responses here with dashed lines. And so similar to the previous research, there was no difference between the two groups in the blood pressure responses to the hand grip exercise protocol. Here are the middle cerebral artery velocity results shown with the solid lines. So women with a history of preeclampsia, again shown in red, they demonstrated lower cerebral blood velocity at baseline and then throughout the entire protocol compared with women who had a history of a normotensive pregnancy. So since the women with a history of preeclampsia had a lower middle cerebral artery velocity at baseline, we wanted to evaluate the middle cerebral artery velocity and mean arterial pressure responses as a % change from baseline. So here again are the blood pressure responses shown as a % change. Again, these were not different between the two groups. And you can see that blood pressure was elevated about 20% above baseline during that last stage of the isometric hand grip exercise. Here are the cerebral blood velocity responses shown as a % change. So again, as you can see, the women with a history of preeclampsia shown in red, they had this augmented cerebral blood velocity response during that final portion of the exercise when blood pressure was the highest. There was no statistical difference between groups during the post-exercise ischemia. And then there was an elevated cerebral blood velocity, again, during the recovery period when comparing the women with a history of preeclampsia and a normotensive pregnancy. So in conclusion, postmenopausal women with a history of preeclampsia demonstrated lower cerebral blood velocity at baseline and throughout the protocol. However, they had a higher % change from baseline during the last third of isometric hand grip exercise when blood pressure was the highest, and also during the recovery period. And this was also true when we added the use of a hypertensive medication as a covariate, a statistical covariate. So these results suggest that there's an effect of the preeclamptic pregnancy history on the sensitivity of the cerebral blood velocity response to a sympathoexcitatory stimulus. And ultimately, these results also support our other studies from the larger project, showing that a history of preeclampsia may influence long-term brain health. And that includes cerebrovascular reactivity to hypercapnia, brain structure, specifically gray matter volume, and cognitive function. So I'd like to end with a final message, and that is being a simple validated questionnaire regarding pregnancy history could be useful in both clinical and research environments when evaluating cardiovascular and cognitive health, specifically when evaluating risk for things like hypertension and or cognitive impairment or cerebrovascular disease. And with that, I would like to thank the co-authors of this study and for allowing me to be a part of this project. I'd also like to thank the Rochester Epidemiology Project, the Mayo Clinic Specialized Center of Research Excellence on Sex Differences, the study participants for their time, and my PhD advisor, Dr. Jill Barnes, and the members of the Barnes Lab, and also our funding sources. Thank you all so much for listening, and I'd be happy to take any questions that you have.
Okay, congratulations. That's a great presentation, and there are already a few questions for you. So from Dr. Rickers, sorry, Carolyn. It was interesting that you saw a difference in baseline MCAV between non-pregnant or normotensive pregnant and preeclamptic groups. What are your thoughts about potential differences in vessel diameter that might account for this effect? Are there any data to suggest differences with advanced imaging approaches?
That's a really nice question. Thank you, Dr. Ricard. So as of now, I am not aware of any data from using MRI or structural imaging to show differences in diameter, especially in the women 35 years post-pregnancy. I'm also unsure if there is data during pregnancy showing differences in middle cerebral artery diameters and differences between those with a preeclamptic pregnancy or a normotensive pregnancy. I will say, however, that in animal models, there is it's been shown that there's amount of vascular remodeling that does occur. And those that have or preeclamptic animal models have less vascular remodeling and actually show cerebral hyperperfusion during pregnancy. So yeah, I think the vessel structure is a really interesting question and point. And as of now, we really don't have information in humans to be able to answer that.
Okay. And so a question from Dr. Broussard. Could a difference in PET CO2 changes partly explain differences in MCAV changes between groups?
Yes, this is another great question, Dr. Broussard. Thank you for bringing that up. So I didn't present the data, but there were no differences in end-tidal CO2 between the two groups. The end-tidal CO2 did not change much throughout the protocol, only 1 or 2 millimeters of mercury throughout the protocol, but there were no statistical differences between the two groups. But thanks for bringing that up.
Okay, great. And a question from Dr. Skow asking about your exclusion of individuals with a BMI above 34. Can you speak to that and how obesity may have influenced your results?
Yes, thank you also for that question. So yeah, that in this particular study, there was not a statistical difference between the two groups in BMI. That being said, in the larger cohort of women studied with these different pregnancy histories, BMI did come up as a covariate. And so in some other analyses, we did include BMI as a covariate, but in this study in particular, we did not. And so that could be something to consider. We just didn't have the statistical power to do it in this particular study.
Okay, that's great. If there aren't any other questions, I do have one. So really great presentation. We know that individuals who have preeclampsia, that they're at very high risk for developing cardiovascular disease. And I noticed that one of your exclusion criteria was anybody who had cardiovascular disease with the exception of controlled hypertension. And I'm sure there's like many reasons for that. But would you expect to see anything different in those individuals who have a history of preeclampsia, who also have subsequent cardiovascular disease?
Yes, great question. So yeah, in this particular study, the women were considered, otherwise cardiovascularly healthy, with the exception of being prescribed or taking antihypertensive medication. So just thinking about in this cohort, some of the data from the brain imaging showed that the effect of having a history of preeclampsia and current hypertension was more detrimental for brain volumes, particularly in the occipital lobe, compared with women that had a history of a normotensive pregnancy or even hypertension, but no history of preeclampsia. So it does seem like there is a potential, at least in that aspect in brain structure, a kind of cumulative effect of the pregnancy history as well as the underlying cardiovascular disease. The other thing I'll mention is also in this group, they found that those with a history of preeclampsia had higher coronary artery calcification. Again, this is in those without this overt cardiovascular disease. So I would say that this group was relatively healthy And so the fact that we're still seeing these differences in cardiovascular and cerebrovascular function, even in these healthy postmenopausal women, is really intriguing.
Okay, that's great. And I'd also like to echo your final slides here in Canada. The Heart and Stroke Foundation of Canada, and I know the American Heart Association, are really advocating for the inclusion of pregnancy history. So for those of you who are listening, strongly recommend this. The word just doesn't get out there. So thank you so much for highlighting that. And thank you for a super interesting presentation. I don't know if there's any other questions for Dr. Miller or any of the other presenters, but if not, then I'd like to thank the organizers for inviting all of us to participate. It's been a fun morning.